Hi, my name is Helen Boltzam. I'm the CEO and co-founder of cellular Biosciences. At cellular, we develop precision gene therapies for the brain. And today, I'm going to talk about how we utilize high-throughput analytics to develop those therapies. Before I get into the analytics part, let me briefly describe the problem we're trying to solve at cellular. We know that most neurological disorders don't impact the entire nervous system, but affect specific cell populations in the brain. For example, Parkinson's disease is caused by the degeneration of a cluster of dopaminergic neurons in the midbrain, and some forms of depression appear to be caused by changes in activity in a group of cells in the subcortical brain structure known as lateral habenula. Although diseases affect specific cell populations, contemporary therapies rarely do. Instead, they engage targets throughout the brain, only a small fraction of which is relevant for a specific disease state. For example, opioid pain medications effectively engage brain's analgesic system, but they also suppress respiration and cause dependence through completely distinct targets in the brain. So the bottom line is that we need high-precision therapies that affect only those cell populations that need fixing. And this is exactly what we're aiming to do at cellular. In this talk, I will tell you how we solve this problem in 3 steps. First, I will discuss how we identify cell populations in brain structures implicated in a disease. Secondly, I will tell you how we identify disease-relevant cell types. And finally, I will outline how to design corrective therapies for target cell types. In this talk, I will focus on pain conditions, but this approach is applicable for a broad spectrum of neurological disorders. For pain, we in fact know about a dozen brain regions that get activated during the pain experience. Pain, as well as chronic pain conditions, stem from activation of peripheral pain-sensitive neurons, and this activation propagates up to higher-order brain centers in the brainstem, forebrain, and cortex. So these anatomical sites serve as promising targets for intervention. However, there is one serious caveat. We don't know what types of cells these brain regions are made up of. For example, one of the midbrain regions that always activates during pain experience also harbors cell populations regulating essential functions like eating, breathing, and body temperature. Therefore, we cannot bluntly modify all cells in the structure. Instead, we need to know what the players or the cell types are and which ones are relevant for our disease state. Let me first tell you how we find the cell cohorts present in the brain region of interest. The technology that allows us to do this is called single-cell RNA sequencing, which enables us to profile all the genes that are expressed in each cell in a particular brain center. And briefly, it works like that. We microdissect out the brain region of interest and process the tissue with enzymes that break it apart into single cells. Then we put it through a microfluidics platform that captures single cells in small reactions, where you prepare a sequencing library from expressed genes from those cells. And sequencing library is essentially DNA that is compatible with a sequencing machine. Next, we sequence the DNA and map the short reads back to the transcriptome. And this procedure yields a gene-cell matrix that is a starting point for our single-cell RNA-seq analysis. The gene-cell matrix tells us which cells express which genes. Rows represent all 20,000 genes that we have in a mammalian genome, And columns represent the cells that we captured through the single-cell RNA sequencing. And usually we have thousands to 10,000, tens of thousands of cells. The goal of single-cell RNA sequencing analysis is to identify cell types in our brain region of interest in an unbiased manner. So I mentioned cell types a few times. Let me now define what cell types are. Cell types are cohorts of cells that are similar in terms of functionality and gene expression profile. To give you an intuition how we define cell types based on their gene expression, let me bring you a simple example. Let's say we want to differentiate cell classes that are clearly different, such as neurons and astrocytes. For your reference, neurons are cells that process information in the brain, whereas astrocytes maintain a chemical environment. And the central concept here is that we can think of cells as points in gene expression space. So these two cell classes are clearly different, and we can differentiate those based on only two genes. A neuronal marker, SNAP25, that is expressed in neurons but not in astrocytes, and astrocytic marker GFAP, that is a gene that is expressed in astrocytes but not in neurons. And when we look at those cell classes in a two-dimensional gene expression space, we can find that these cells form two clearly distinct clusters or communities. And note that the x-axis depicts the expression of the neuronal marker, and the y-axis depicts the expression of the astrocytic marker. And as you can see, astrocytes cluster in the upper left corner because they express a high level of GFAP but no SNAP25. And neurons cluster in the lower right corner because they express a high level of SNAP25 but no GFAP. So these cell classes are easy to identify in a two-dimensional gene expression space. Well, in reality, we don't know the genetic programs that define cell types, nor do we know how many different cell types there are in a brain structure. Therefore, instead of 2 dimensions, we have to identify these cell types from 20,000 dimensional space, as there are about 20,000 different genes in the mammalian genome. So how do we find those cell communities in this high dimensional space? So the input data for our analysis is the gene cell matrix, where the values are gene expression levels in a given cell. It turns out that we do not have to look at all 20,000 dimensions because there are a bunch of genes, so-called housekeeping genes, that are expressed in pretty much all cells. And these are genes that are required to maintain the health of every cell. Because those are expressed in all cells, we can ignore those because those don't really help us differentiate cell types. So we can threshold those out as low variance genes. Instead, we focus on analyzing genes that are expressed in a highly variable manner across the dataset. In other words, we want to focus on genes with high expression levels in a subset of cells and with low or no expression in others, because those are most likely informative, determining how cell types differ from each other. As the first step, we compare dispersion of genes and filter out the least variable genes from the dataset. Usually, this leaves us with from a few hundred to up to 2,000 genes. At this point, let's assume our cells reside in a 1,000 dimensional space. It turns out that many of those highly variable genes are highly correlated, which means that we can project this high dimensional data set into a lower dimensional space without compromising the information content. To that end, for the purposes of dimensionality reduction, we use principal component analysis, or PCA. The goal of BCA is to find a lower dimensional representation of the data so that it captures as much of the information of the original data set as possible. It finds principal components of the data set where there is most variance in the data. The first principal component is a linear combination of the features that has the largest variance. The second principal component captures the second largest variance such that it is orthogonal to the first one, and so on. In practice, that leaves us with about 10 to 25 dimensions that captures most of the variability in our data set. And as a reminder, in our now 15 or so dimensional space, the dots represent cells, and we need to find communities of cells that represent cell types. In terms of what we're trying to find, the logic is that a cell type is a cluster of cells that is close together in the principal component space because of the similarity of gene expression pattern in a specific cell type. For the purposes of finding cell type clusters or cell communities that are transcriptionally similar, we use shared nearest neighbor modularity optimization-based clustering algorithm. We start by creating a graph-based representation of our dataset where cells are nodes and cells with similar expression patterns are connected with edges. So first, to construct a k-nearest neighbors graph and use Euclidean distance to determine k-nearest neighbors for every cell in this principal component space. Then we refine the edge weights of the graph by calculating the neighborhood overlap or Jaccard index between every cell and its nearest neighbors. We construct the shared nearest neighbor graph by thresholding out lower weight connections. And the reason we convert our data to a graph-based representation is because there are efficient and really elegant algorithms for clustering or identifying communities within these graphs. And one of those algorithms is Louvain method that clusters cells by optimizing modularity. At this point, our data is still a 15 or so dimensional space. So to visualize the results in a two-dimensional space, we're using a TSNE embedding or D-distributed stochastic neighborhood embedding. This is a nonlinear dimensionality reduction algorithm that projects cells from the principal component space into two dimensions such that similar objects are modeled by nearby points and dissimilar objects are modeled by distant points with high probability. So cells that are transcriptionally similar cluster together and cell types that are dissimilar cluster further apart. And we color code the identity of cell types based on the results of the shared nearest neighbor algorithm where each dot is a cell. Now I have walked you through the process of identifying cell types in single-cell RNA-seq data. The value of this analysis is that it both tells us what the cell types are and reveals the genetic programs that define those cell types. Here I'm showing a heat map of cell type-specific gene expression. Each row is a gene, and each column is an individual cell clustered into cell types. Gene expression level is color-coded such that warmer colors represent higher expression. And as we can see, each cell type is clearly identifiable by a dozen or more locally uniquely expressed genes. This is important because these programs enable us to recognize those cells, and as I will tell you later, will also give us a way to control those cells. So the bottom line is that each cell type has about a dozen uniquely expressed genes that differentiate them from the rest of the cells in this structure. Now I have told you how to identify cell types in a brain region, and the question is, why is this relevant? Well, it turns out that specific cell types mediate distinct functions. For example, let's look at cell types in dorsal root ganglion, which is a cluster of cells that enables us to feel touch, temperature, pain, and other sensations. In dorsal root ganglion, we actually happen to know which cell types mediate which functions. On the right, I'm showing you color-coded cell types that mediate distinct stimuli. For example, green cluster on the right mediates mechanical pain, something you would experience when you hit your toe. And the red cluster mediates pain caused by high temperature. And selective manipulation of cell types can control behavior. Let me demonstrate that with two videos. So in this video, we have a regular mouse whose paws are stimulated with blue light. And as you can see, the mouse is completely indifferent when the light is directed to his skin and paws. And this is because the blue light is completely harmless. In the second video, I'm showing a mouse where we have rendered heat pain sensing neurons blue light sensitive, such that exposure to harmless blue light triggers heat sensation. So this mouse feels high temperature on his paws and skin. And importantly, this sensation is caused not by heat, but by the activation of the cells that mediate high temperature. This triggers paw shaking and licking, which are typical care behaviors after sensing heat. So these two videos demonstrate that we can control behavior by activating certain cell types in the brain. Similarly, if we know which cell types cause specific neurological disorders, with the right technology, we can manipulate those cell types to manage or cure the disorder. So the question is how to go from cell populations to mapping functions to those cell types. Well, this used to take years of research. There are now fast and efficient ways to map functions to cell types. One such approach takes advantage of the same single-cell gene expression profiling that I described before and relies on detecting transcriptional signatures that accompany alterations in neuronal activity or disease states. For example, if we wish to identify neurons that participate in pain processing, we would microdissect out the brain region implicated in brain processing in control mice that have not experienced a painful stimulus and a test group that has. We would dissociate the tissue and perform single-cell RNA-seq. And after we identify cell types in these brain regions, we can just visualize the expression of genes that are upregulated upon neural activation. And as a side note, these activity-regulated genes are known as immediate early genes, or IEGs. And we can just plot their cell type averaged expression for each cell type in control and stimulus cases. So, and in this toy example, we would not see any expression of these IEGs in a brain region of interest in the control mice. However, exposure to different stimuli elicits a distinct pattern of activation by cell types manifested in IEG expression in neurons that are activated by a given stimulus. In practice, this data looks something like that, and I'm showing you expression of one of those immediate early genes on the y-axis, and on the x-axis I'm showing you cell types in one of the brain regions. So there is no expression of immediate early genes in the control group. However, if you apply stimulus, you can see that a single cell type strongly upregulates the expression of immediate early gene, revealing which neuron type might be relevant for controlling respective behavior. So if this stimulus is pain, then the pain-activated cell type constitutes a potential target for pain therapies. Now, as we have found all cell types in the brain region that we are interested in, as well as determined the cell type to target with a therapy, we need to figure out how to manipulate those dysfunctional cells. At Cellular, we use gene therapies for this purpose. On a very high level, gene therapies work by introducing new genes or eliminating harmful ones from the cells of dysfunctional organs with a goal to restore their functionality. But one thing to notice is that today's gene therapies work well for simple organs, like the liver, where we have only a few distinct cell types. However, they fall far short for more complex organs, such as the brain, where we have about 2,000 distinct cell types. And the challenge is that currently available gene therapies lack the precision to distinguish between different kinds of cells. At Cellular, we've developed a patent-pending cellular engineering platform called CellSelect that allows us to gain selective control over any cell type in the brain by harnessing the genetic programs that define the cell. So how does this work? In the first part of the talk, I showed you how we can identify gene expression profiles that define individual cell or neuron types. Here we take advantage of these cell type specifically expressed genes to express therapeutic effector genes that either activate, inhibit, or kill cell types that need fixing. And to achieve that, we use the CRISPR-Cas9 system to program a targeted knock-in of the therapeutic gene. And CRISPR-Cas9 is the genetic scissors technology for which they issued the Nobel Prize a few weeks ago. We do this knock-in by targeting the genomic locus that is expressed in a cell type specific manner. Depending on what the therapeutic gene is, the target cells will either be activated, lesioned, or silenced. On a practical level, we use adeno-associated viral vectors or AAV vectors to deliver the CRISPR reagents and the therapeutic gene to the desired brain region. And the anatomic location is targeted through a one-time guided brain injection that delivers the AAV vectors. And indeed, if we deploy this viral vector-based gene therapy in mice, we can achieve a high targeting efficiency of the desired cell population in live animals. Crucially, this approach allows us to re-engineer central nervous system targets that are not pharmacologically precisely targetable, but through our technology are precisely re-engineerable by gene therapy. And in our ongoing work, we are deploying this technology to re-engineer the central pain-mediating cell types with an aim to provide effective therapies for different chronic pain conditions. And another disease-focused area we're pursuing are focal seizure conditions. In conclusion, I've covered three things. I've shown you how single-cell gene expression profiling reveals cellular diversity in brain regions of interest. Then I demonstrated how single-cell RNA-seq based stimulus to cell type mapping identifies therapeutically relevant cell types out of all cell types in a brain region. And finally, I've described an avenue for a precision re-engineering of central nervous system cell types for therapeutic applications. And with that, I would like to close. Thank you, and I will take any questions.